Autophagy plays an important but underappreciated role in shaping the health of the gut and its microbial inhabitants, and this connection is increasingly recognized as critical in the context of obesity. The gastrointestinal tract is not only responsible for nutrient absorption but also serves as a major immune and metabolic interface between the body and its external environment. At the heart of this interface is the intestinal barrier, a layer of epithelial cells tightly joined together, lined with mucus, and inhabited by trillions of bacteria. When this barrier is compromised, it allows microbial fragments and toxins to leak into the bloodstream, a process called metabolic endotoxemia, which fuels low-grade systemic inflammation and contributes directly to obesity and insulin resistance. Autophagy helps prevent this scenario by maintaining gut barrier integrity, modulating immune responses, and even influencing the composition of the microbiome itself. One of the ways autophagy supports gut health is by clearing damaged epithelial cells and supporting the renewal of tight junction proteins, which help seal the intestinal lining. When autophagy functions properly, the gut maintains a selective barrier, permeable enough for nutrient exchange, but robust against pathogen invasion and toxic leakage. However, in obesity, autophagy in gut cells tends to be suppressed due to chronic nutrient overload and persistent inflammation. This leads to dysfunction in the gut lining, allowing lipopolysaccharide, LPS, a bacterial toxin, to enter circulation. Even small increases in circulating LPS can activate immune responses and insulin resistance, worsening metabolic outcomes. Moreover, autophagy is involved in controlling the balance of immune cell function in the gut, such as macrophages and dendritic cells, which monitor microbial activity and respond to pathogens. Impaired autophagy in these cells leads to exaggerated inflammatory signaling, contributing to a pro-inflammatory state that not only disturbs microbial communities, but also promotes obesity-related complications. This dysregulated immune environment reinforces microbial imbalances, favoring the growth of pro-inflammatory bacteria and reducing the presence of beneficial, short-chain fatty acid (SCFA), producing species that normally support metabolic and gut health. Excitingly, this gut autophagy axis is not a one-way street. The microbiota also produce metabolites that directly influence autophagy in host tissues. For example, butyrate, an SCFA produced by fiber-fermenting bacteria, has been shown to stimulate autophagy and improve insulin sensitivity. Thus, supporting a healthy microbiome through dietary fiber, prebiotics, and fermented foods can promote autophagy in the gut lining and elsewhere, creating a reinforcing loop of microbial and metabolic health. Likewise, improving autophagy through fasting or exercise can help restore gut barrier function and promote a more balanced microbial ecosystem. The interplay between autophagy and gut microbes also holds promise for therapeutic development. Interventions that mildly stimulate autophagy, such as calorie restriction mimetics, certain polyphenol, or probiotics, could help normalize microbial composition and reduce gut-derived inflammation. These approaches not only benefit gut health, but may also reduce cravings, improve fat metabolism, and help modulate energy expenditure, all of which are central to sustainable weight management. Still, there are complexities that need attention. Overstimulating autophagy in certain gut conditions, such as inflammatory bowel disease or severe dysbiosis, may backfire if not carefully managed. In these cases, targeted or cyclic activation may be preferable to continuous stimulation. Precision nutrition and personalized microbiome analysis could help tailor these interventions for individual needs and microbiota profiles. Ultimately, the relationship between autophagy and gut microbiota adds a critical layer to our understanding of obesity. It shows that metabolic health is not only a matter of calories and hormones, but also a question of microbial ecology and cellular housekeeping. Enhancing autophagy helps restore the gut's role as a protective, intelligent barrier and nurtures a microbiome that works with, not against, the body's efforts to maintain balance. By nourishing this relationship, we open the door to more holistic, effective, and sustainable strategies for preventing and reversing obesity.